Thank you very much. And we can, in fact, we'll give our mentor a round of applause. Everyone's getting claps today. Thank you very much. But if you have questions, comments, concerns just about this class in general and in the future, feel free to turn to Kenji and a few other of the peer mentors we're going to have. So, just a bit of housekeeping advice here at the beginning. Some due dates for your homeworks and team logs. If it is due in your discussion section, it'll always be due that Thursday. These are things like your any kind of group work you do, so your team logs, your I don't know, any sort of documentation. That's going to be due on Thursdays. Homeworks, tests, things like that are always going to be due on Wednesdays. They're going to happen on Wednesdays. For the submission of your homeworks and team logs and whatnot, it seemed like Canvas threw up a couple errors saying that you couldn't submit it online. I hopefully have clicked the boxes that you need to do that. Have you gotten a chance to check?
Yeah.
Okay, great. My apologies for any confusion there. Canvas is a fickle beast. It's
a monster.
And your homework will actually be posted on Monday. It'll be due about 10 days after that. So not the Wednesday that followed, but the Wednesday after that. Everything that you're going to need to know for that homework will be taught a week in advance. So the final thing that'll kind of be on that homework will be taught on that Wednesday. So that's just a bit of the housekeeping details. Any others from the sides of the room?
Sure, don't forget there's another review session tonight that I'll be running again. Again, it's not required. If you want to come, great. Don't hesitate to. Remember, you have other resources. You have the three of us, because we all know the chemistry very well here. You have other teammates on your team that might do a bit with you individually. There's the optional biology textbook that we've got. You have your old textbook from high school. That's another resource as well. There's also something called the Khan Academy. How many people have heard of the Khan Academy? It is a really cool resource. You know, I'm very skeptical of a lot of stuff online. It's just, let's face it, 99% of what's on the web is garbage, right? But this is actually really good. In fact, those of you who are going to med school, I know that includes a few of you, if you're preparing for the MCAT to review your basic sciences, often enough people recommend you go through some of those Khan lectures, differing to 10 minutes, in preparation for your MCAT. So that's another resource you could use to blow up on this. Let's see. And don't forget, tomorrow, Thursday, as Dr. B mentioned, you've got your first log and discussion. And also, there should be a notice already online about where that discussion section is. It's going to be in a computer lab because we have to combine two sections all at once. And it's basically a preview of the lab, but also anything that you need to know about the prospectus we'll be talking about there too. So once you shouldn't have to do any more research about the prospectus. Once you take it, sat in and paid attention to what we're doing tomorrow in discussion. Any other comments or questions about it? Thank you very much.
Okay, well, thank you very much. To the folks in back who clearly have computers that really ought not to, I would recommend that you close them. You can keep them up, but it's mostly to your own detriment, just because you won't learn as much as the rest of us. So, a note on your projects. So I've gotten a few, oh yeah, probably help if I connect this thing. My goodness. All right, a note on your projects. So I've gotten a few questions about what are these projects kind of about. And so to give you some sense of this, I just wanted to give you the titles of last year's projects. These are things like a myotrophic lateral sclerosis diagnostic system. You've got a papillary thyroid cancer urine diagnostic test. Using immunocytochemistry to diagnose esophageal cancer. If you don't understand all these words just yet, that's okay. This is really more to give you a flavoring. In fact, I had to go through about a decade's worth of biomedical engineering experience to be able to identify a few of these myself. When I sat in your chairs, I did not know any of these words. And in fact, standing up here to this day, I had no idea what any of these words mean. I didn't know what idiopathic pulmonary fibrosis is. This is all to say that biomedical engineering is a big field with a lot of words, a lot of ideas associated with it, and we're going to kind of learn as we go. We'll accumulate all this knowledge, both individually and as a group. But that also means you need to be mindful when you're talking to other people of their level of technical competence. So when you start talking to your clinician, or you want to start pitching this to entrepreneurs, or you just want to tell your mom, dad, sister, brother, aunt, uncle, neighbor, coworker what the heck you're talking about, this is really what they're getting out of it. They're going to recognize every now and then a word. They're going to know what cancer urine diagnostic test, sort of what each of those words individually mean. They might not know what your big picture is trying to get at. So when you start talking to people in technical language, just be mindful of what they know, what you know, and what you might need to explain along the way. And that's just something to keep in mind as you go ahead in the project. All right, so today's lecture is an introduction to cell and molecular biology. And our objectives today are to understand a few key parts of the cells, key parts of the cell and their functions. So there's going to be a few things we really need to know about basic eukaryotic cells and what those things do. We're also going to kind of want to know from a systemic approach how this all fits together. Cells and their constituents have systems and subsystems that all correspond to a cellular systems model. And we're going to want to be pretty familiar with the inner workings of that. And then finally, we're going to want to start to appreciate how all these systems come together, how these systems and subsystems come together so that we can develop a diagnostic test. Because that's not only ultimately the goal of this class in terms of the project, but it's what we want to be able to do as biotechnologists, as biomedical engineers in general. So be sure to always keep your eye on the prize. All right, so a cell, so far as I can tell, looks a little bit like this. This is probably something we're all familiar with, and if you've heard of this material, if you feel comfortable with this material, that's okay. I'm basically trying to take your entire high school education's worth of biology and cram it into about an hour. So we're going to see what we can do. So does anyone want to venture a guess of, say, what this thing right here is? Does anyone? Okay, so one at a time, maybe. No, actually, keep shouting, keep shouting. What made you recognize that as the mitochondria?
Looks like it.
All right, maybe we. All right, so I saw a hand up, Ryan.
Because it has its own membrane and it has like folds within it.
It's got a membrane. It's got its folds. Anyone got any other? Yes, Anna.
Looks like an M.
It looks okay. Maybe that was the giveaway. A little mitochondria, some little there. Any other things? No, I mean that's a great reason. There is a pretty unique structure and look to the mitochondrion. And mitochondrion is the singular of mitochondria. I'm just going to go ahead and keep saying mitochondria the same way we all say data instead of datum when we're supposed to. So just a thing to be in mind. Does anyone? Oh, darn it! Does anyone know what a peroxisome or a peroxisome is and why it might be so closely situated with the mitochondria? Yes.
Break down like oxygen, like bad particles.
You got all the right words in something of the wrong order, but that's close. Let's see if Nighty can help.
Breaks down nitrogen peroxide.
Again, you're close. You're right. You're entirely right. It basically takes toxic molecules or long fatty acid chains and breaks them down using hydrogen peroxide, things like that. Yeah, entirely correct. Does anyone know what sort of the jelly is of the... Yes, Emily.
Cytoplasm.
Cytoplasm, we also call it the cytosol. Yes, cytoplasm, cytosol is basically what all of this happens in. It's salty jelly. Does anyone know what this little funky, say, apparatus might be? Okay, we got the Golgi apparatus. That's entirely right. It's, I believe, Dr. S's favorite organelle because it's the only apparatus in the whole place. So that's a good thing. It's also called the Golgi complex, the Golgi body. It's, in fact, I think the only remaining organelle still named after a person, a fellow named Golgi. Closely associated with the Golgi apparatus is something right here. Does anyone hazard a guess? It connects, yeah, all the way in back. That's the endoplasmic reticulum. Entirely right. I don't need to teach any of this class. You already know it. Fantastic. Associated with the Golgi apparatus and the endoplasmic reticulum are these little budding features. Does anyone know? Your hand shot right up before I even finished the question. What is that little budding?
You like the sickles?
Yeah.
They're reticulums.
Those aren't ribosomes. That would be a gigantic ribosome associated with the endoplasmic reticulum. And ribosomes are really small, but I'm glad you know that is associated. Let's see if we can hear from someone else. I know you know it. Anyone got a guess? Anyone got a guess? All right, Knight is going to tell us. That is a vesicle, entirely right. A vesicle is basically a budding off of a membrane to transport materials. Oops. Well, it looks like we now know what the nucleus is, but I bet you could have guessed that big central thing. Does anyone know what surrounds the nucleus, this little thing around it?
Nuclear.
Okay, way in back, Sam.
Nuclear membrane.
The nuclear membrane, the nuclear envelope, entirely correct. This controls the passage of, this controls the flow of material into and out of the nucleus. Yes?
Do vesicles only bud around the endoplasmic reticulum or different organelles too?
They do it mostly around the endoplasmic reticulum and the Golgi apparatus. Does anyone know why? Why that might be the case? Okay, right here and back.
So the Golgi apparatus packages things inside vesicles.
In case you couldn't hear that, he basically said the Golgi apparatus packages things into vesicles to transport them across the cell. So that's basically the whole point of a vesicle, is to get it from one place to another. This whole lesson's going to last about 15 minutes with it. Does anyone know what sort of surrounds the cell? Cell membrane, yeah, that's a gimme. And then finally, we got a special kind of vesicle up here. It's got a couple little bubbly features. Does anyone know a special kind of vesicle? All right here. A lysosome, yes, exactly. And what does a lysosome do? It digests things, exactly right. Fantastic. Okay, so this is basically all the components of the cell you're going to need to know in this class. You're going to need to know how they all come together, why they're all important, but it doesn't always look like that. This is a cell. Does anyone think they could come up here, with the exception of the nucleus here, and point out any of the things we just mentioned?
Cell membrane.
All right, y'all got the cell membrane. Okay, but that's great. You got the cytosol. All right, we got a couple of jokers in the class, but you're entirely right. You can identify a few things. The mitochondria appear to have some of that weird structure we were talking about. We can identify a few lysosomes, things like that. But when it comes to actually identifying real materials in real cells, we got to be mindful that it doesn't always just look like that diagram you've got printed out. That this is actually the sloppiness of biology. But it turns out the idea of a cell is part of a thing we in the scientific community call the cell theory. And a theory is not a guess, it's not a hazard, it's not a hypothesis. A theory is a collection of knowledge, facts, tested and proven or failed hypotheses that come together to form a framework upon which we can drape everything we know. And the cell theory has three key components. The first is that all living things are made of cells. We basically have defined that anything alive is made of a cell. That almost definitionally leads to the second part of the theory, which is that the cell is the smallest living thing that can perform all the functions of life. So now I'll give you a moment just to write that down. So all living things are made of cells. The cell is the smallest living thing that can perform all the functions of life. And then finally, the cell theory posits that all cells must come from other cells, from pre-existing cells. All parents need parents of their own. All children need parents. All parents need grandparents. All this is to say that a cell must produce, a cell must be produced from another cell. I'll give you just a few more furious handwriting notes there. That's great. This is the sort of thing. Now, does anyone... I don't need to ask this question. The reason we care about cells in particular, and especially in this class, is ultimately they are the smallest living thing that can perform all the acts of living. When we talk about treating patients or populations or fixing tissues or engineering them, we're ultimately talking at bigger and bigger scales of cells. Everything is happening finally at the unit of a single cell. So this is the important part of what constitutes life. But then I pose to you, what then is life? Here's a philosophical question for you. Does anyone have a good definition of what life is? Yeah, way back, Sam, our first philosopher.
So one, it has to
Okay. We've got the celibate as not living. Okay, that's great. Okay, let's see if we can add a few more details to this. All right, here and back. Has to have metabolism? Okay. Got metabolism. If on your tombstone it says he metabolized, will you feel you've lived the life? All right, great. Let's see, Alan.
I don't know if it's too vague, but basically like expend energy or have some sort of like energy.
That's kind of metabolism. Expend energy. All right. He spun his wheels as he lived. Yes.
Maintain homeostasis.
Maintain homeostasis.
That's the first one.
All right, these are a few pretty good details. I know, there's a few others. Oh, you're keeping your hand up. Okay.
Not be dead.
Not be dead. We can define it by its inverse. Not being.
I'm sorry, I had to get that joke in.
Well, then, unfortunately, you have to answer the follow-up. What does being dead mean?
Not being alive.
Tautologies will not help you in this class. Let's see if, here's a few definitions that I found in the literature for what defines life. None of these is 100% correct, but they are a pretty good stepping stone. So life, stated as such, is the quality that distinguishes vital and functional beings from purely chemical matter. So standing in front of you is a man made of oxygen, phosphate, nitrogen, some salt water, but I'm animated. That sort of, distinguishes me from the dead. Purely chemical matter. Number two, oh goodness, I didn't mean to gray that one out for you. It is the state of a material complex or individual characterized by the capacity to perform certain activities. So this is what a lot of people were getting at here. Metabolism, growth, reproduction. We had maintain homeostasis instead of growth, but otherwise we nailed metabolism, and Sam had his priorities straight with reproduction. Finally, this third one we've got is a sequence of physical and mental experiences that make up the existence of an individual. These are a few ways we can start to think about life. But as we know, it's a little more than that. It's sometimes sitting in class bored or excited about what's coming next. It's the rush you get listening to your favorite song, talking to friends. It's so much more than just some of these words. So as you go through the semester, do see if you can come up with a definition for life that satisfies you. Because the true calling of every engineer is to be a good citizen of the great society, living the noble life. So try to do that. But let's see if we can try to do that at a few different scales. Because life doesn't just exist at the cell level. It exists a number of different places. All the way from, say, the living being you see in front of you, or blue whales, or hamsters, or amoeba, all the way down to atoms. Things like this all matter to the functioning of life. Some of the sort of key things that we're going to want to be focused on is that cells tend to exist from about 200 microns. Is everyone familiar with the concept of a micron or a micrometer? Anyone not familiar? That's okay. You're hopefully all at this point going to become familiar with SI units. But a millimeter is just 1,000th of a meter. A micrometer is just a 1/1,000,000th of a meter. I'm going to say micrometer and micron kind of interchangeably. But cells exist from about 200 microns till about 0.1 micron, or about 100 nanometers. Organelles, rather, that is the constituent components of cells, tend to exist a little bit smaller than cells, from about 2 microns-ish to maybe about 20 nanometers there. Things such as ribosomes tend to be some of the smaller organelles that we've got. And then finally, molecules kind of exist just at the hair's breadth of an organelle and smaller. These are proteins that might get delivered for, to regulate your blood sugar, things like plasma. All these sort of molecules exist in usually the nanometer range and below. When we had seen that picture of the cell, what we had resolved it with was in fact a light microscope. That picture of the cell was taken with an optical microscope. And there's actually a limit to each of the ways we can resolve and look at each of these materials, the way we can study them. Things kind of in the higher region here, but we'll just call it Middle Earth, are what we as human beings can resolve. We understand things kind of middle in size. We don't understand the whole shape of the cosmos or down to the atom, but we kind of understand a hamster and about how fast it goes. We also understand about the middle range of visible light. We don't know infrared or ultraviolet. And we know things kind of going at middle speeds, so nowhere near the speed of light and nowhere near the speed of molasses. We kind of understand things in between. Sort of below that is what is resolvable by a light microscope. You can get down to about 200 nanometers with a light microscope, but not much further. That's because light, visible light, has a wavelength associated with it. And wavelengths themselves define the resolving power of microscopes. So if we want to get a little bit smaller, we have to shear the visible electromagnetic spectrum into the invisible one with an electron microscope. Things like scanning electron microscopes and transmission electron microscopes. These are what can see molecules and some of our smaller organelles. For this particular class, we're going to be interested right here in this middle region, from basically the size of cells to their constituent components. This is where we're going to concern ourselves. So a lot of the pictures we're going to see might be from optical microscopes. Okay, what then separates life from non-life? And I'm looking for something of a very literal answer here. Does anyone have a guess? You already said the living and the dead. Does anyone hazard a guess? Anita, your hand went slightly up. I'm very sorry. You got to answer now.
I think something that can't, something that doesn't do like the certain activities that you said, like the producing.
You're going a little more philosophical than I need. I mean, literally, what separates it? If you take a look at this picture, what's the thing separating the living from the non-living? All right, way in back. Let's see if Greg can help.
Conducting life processes.
Oh, you're all going. All right, here's how literal I meant. The cell membrane. The cell membrane is literally the thing separating life from non-life. We need to know a little bit. And in fact, I think some of you do have your notes. So some of you were ringers maybe, but you probably got where I was going. So the cell membrane. The thing you need to know if you know nothing else about the cell membrane is that it is made of phospholipids, basically little tiny things that all come together in a fluid mosaic pattern to give the cell its shape, its flexibility, and a whole bunch of other things. Now, these phospholipids have a head group and a tail group. And we can kind of look at these a little more specifically. In the head group, and in fact, we're dealing in this case with a very specific kind of phospholipid known as phosphatidylcholine. This is what typically makes up a lot of your cellular membranes. And it's got a polar head group, which is hydrophilic. It likes water. In this case, it's made of choline and phosphate. And then it's got a non-polar or hydrophobic tail. And there are actually two tails, one of which has a kink. Does anyone know why phospholipids have that kink? What is the ultimate reason? Why would we want a kink in our tails? All right up front.
Maybe if they were straight, there would be nothing to link them together. They would just sort of be smooth against each other, whereas this would kind of make it like Velcro.
Kind of like Velcro. That is a great observation. That is one of the reasons. They kind of have to bump into each other, in case you didn't hear it. They kind of need to bump into each other to sort of keep their shape. They need to be touching each other and overlap. But we might be able to accomplish that if, they were just kind of going out at an angle. So why a kink? Anyone got another guess? We got one here in the middle. That is the proximate reason. Yes, you're entirely right to notice that. In fact, the difference here is that there is a double bond. If you haven't yet taken your chemistry course or don't remember, a double bond is just a kind of bond. You can have a singular bond or a double carbon bond. That gives it a little bit of a strength and changes the actual angle of inclination. But why, pray tell, after billions of years of evolution, have we settled on a kink? It's okay. Yes, all right here in back.
It's like allow them to slide next to each other.
Allow them to slide next to each other is a fantastic answer. In fact, if you think about the cell membrane, it's a big undulating structure with mostly in a spherical spherical orientation, and it's being subject to a lot of different stresses and strains and movements. And in order to compensate for the stress and strain, because being in a sphere is actually one of the worst things you can do for yourself. So, try not to ball yourselves up too much. But by allowing there to be a kink, you introduce a little bit of elasticity to the membrane. And that elasticity allows the cell to keep its shape or undergo deformation without bursting. So this is why they want to stick together, and they also don't want to break apart. So this is why evolution has settled on giving that tail a little kink. Yes?
Elasticity like a springing?
Like a springing, exactly right. In fact, we pretend, when you look at a model like this, we are pretending or not telling you that these tails are moving all the time. Not only are the tails sort of moving, but each of these bonds are themselves flexing. But this little double bond basically allows a whole nother bit of levering action to happen. And that all the energy required to push it down is stored as potential energy, which is the basis of elasticity. Yes?
So, does that mean that the double bond then is like more flexible than the single bonds?
Not necessarily. That's a great question, and it's actually hydrocarbon specific. I don't necessarily know. I can look into that if you'd like, if it's more flexible, but it's not, it wouldn't be right to say that a particular bond is flexible in this particular case, but rather the whole structure being itself flexible. In general, the double bond, I think, if I recall from chemistry, is a little bit stronger. Do I have that right?
It is strong.
Yeah, it's a stronger bond, so you're less likely to bend at the double bond, but you do have a whole bit of structure that can now have more space to bend in general in the hydrophobic region. So great question, great question. Okay, so that's basically the basic unit of our phospholipid bilayer. We had talked about this. They assemble their hydrophobic tails towards one another and all their hydrophilic tails, or head groups, towards water and cytosol. There's also things called glycolipids, which are lipids that have been tagged with a sugar or a glycogen, a carbohydrate of some sort. And this is particularly important in being able to identify a cell or signaling in a cell. Other things that exist here in the cell membrane are a bunch of different kinds of proteins. So one of the most important proteins as you go through your career to identify are transport proteins. Proteins that allow ions and other materials to go from one side to the other. There's passive diffusion of materials. There's active diffusion, such as sodium and potassium pumps to pump to make sure we can keep our electrical potential on either side. And these span the whole cell membrane so that material can be transported from one side to the other. And usually when we talk about material in this case, we're talking about very small molecules, usually ions. Just like their glycolipid cousins, glycoproteins are proteins that have been tagged with a sugar group, again for identification purposes usually. This can help you, again, if you're designing a diagnostic test, as being able to identify cell-specific glycoproteins. A lot of cells, or a lot of proteins will also take on what's called the alpha helix structure. You'll find in biology that the helix is one of the most stable structures that exists. And in proteins, this is known as the alpha helix. This isn't a particular kind of protein, but rather a protein structure. And many types of alpha proteins serve many types of roles. For instance, also aiding in the mechanical strength or elasticity of the cell membrane. You've got, we can define sort of a few other different kinds of proteins. Surface proteins, which are proteins that just bind to the head groups of the hydrophilic head groups of the phospholipids. You've got integral proteins that go through the whole way, which means halfway through you know they have a hydrophilic region, otherwise they couldn't do that. They would be repelled in the middle. And then you've got peripheral proteins, which tends to suggest that one side of it is hydrophilic while the other side, or one side of it is hydrophobic while one side of it is hydrophilic. So these are just some of the different kinds of proteins you're going to want to be familiar with as we go forward. Then there's a whole bunch of other stuff. So there's cholesterol, which I'm sure many of your father's uncles have complained about. There are carbohydrates that will inundate the cell. And then there are cytoskeleton filaments. But since we touched on the elasticity of the cell membrane, does anyone know what cholesterol does?
It keeps the either fluid so they can move all around.
That's a great way. Great way of saying it. It keeps it fluid. It actually provides an ordering. Because what we don't know, or what we haven't told you, is compared to the tail groups of our phospholipids, cholesterol groups are straight up and down. They are straight molecules. So that means when you get a whole bunch of them together, they don't allow these tails to start flexing in and out. They, in fact, order the entire thing and form lipid rafts, which allows the fluid mosaic model of the cell membrane to exist. So cholesterol is basically an ordering molecule. And so when you have too much of it, you might in fact make your cell too ordered. And that means the radius of curvature of your cells can't be maintained, and they will lyse, and that's not good. Ask any of your friends with high cholesterol. Okay, so now that we kind of know all this stuff in the cell membrane, how does all of it get there? All right, Emily, what do you think?
Well, when the organism or cell or whatever, it takes in food or molecules from the outside world, and then it gets it in there, and then somehow gets it into the cell membrane, when it eats it, basically.
I suppose I'm looking for a little more detail than that. That's great. That's entirely right. We do eat stuff. It does end up ultimately sometimes on our cell membrane. But in this case, what I'm looking for is we got some roadways. We've got some cytoskeletal structures that deliver all these things to that cell membrane. And does anyone recognize, besides a few ringers in this class that I've already told, does anyone recognize these cells? All right, we got a guess right here in the middle.
What kind of reminds me of neurons?
We got neurons. These are not neurons. Good guess. One of the reasons you could tell they aren't neurons is they actually don't have a huge dendritic structure. But that's a great guess. All right.
I was going to guess HeLa cells.
They are, in fact, HeLa cells. And if you have not read the book, like I think you were required, I would recommend it. It is very good. Show of hands, again, in the quiet of your own heart, you'll know if you're lying to me when you put your hands up. But how many of you have read the book? All right, fantastic. If you haven't, maybe take it as a recommendation from the rest of your cohort. These are HeLa cells. They have vast bioethical implications. They have helped save millions of lives. They have invaded the privacy of one woman, the whole story of which could be told by Rebecca Skloot in her excellent book, The Immortal Life of Henrietta Lacks. We're actually also going to be dealing with these cells in this lab. So become prepared. But the actual thing, the actual cytoskeletal structure here, is this sort of cyan color. Here in the purple-blue sort of is the nucleus, and tagged over here are some of the actin filaments, which are a kind of cytoskeletal element. This makes them nice and sticky. Okay, so we've got ourselves a cytoskeleton, and the cytoskeleton, yes?
Sorry, those are multiple cells, right? And when you said nucleus, you meant they each correspond?
They are each a nucleus, yes. That is a great clarification. Yes, each one of these is a cell. One cell, one cell, one cell. This is all individual cells. Great clarification.
They're just all kind of near each other.
They're all kind of near each other. As you will notice in your class, HeLa cells all kind of clumped together. Okay, so we've got ourselves a cytoskeleton. And the cytoskeleton itself has, I think you might know in your notes, three main components. The first are actin filaments, the ones that I want you to know. These have about a 7 nanometer diameter, and they are critical for things like cell motility, which is the movement of cells. In fact, while you're writing that answer down, you are in fact engaging your actin filaments. They are important for things like endocytosis and exocytosis, which we'll get to in a minute, and for cytokinesis. Does anyone know what cytokinesis is? All right, in back we got a hand shooting up. Exactly right. It is when the cells reproduce and expand. M phase, anaphase, all those things you probably had to color at some point. That's precisely what cytokinesis is. So the filaments involved in that are actin. Now, a few drugs that you might want to know that affect actin filaments are I forget how to say this, cytochalasin, I believe is how it is, actually caps those filament ends, and it prevents the polymerization of the materials, because those actin filaments are constantly polymerizing and depolymerizing, because they basically need to add to each other and expand based on the needs of the cell. Similarly, latrunculin binds the monomers. So rather than capping the filaments, it actually takes each of the actin monomers and binds to them. And that's another way of stopping polymerization. And then phalodin binds and stabilizes filaments. Now, just in general, the cytochalasin is the only one you might experience in the clinic. Has anyone heard that term or know where you might use it? In fact, you might not even know where you'd see it in the clinic. Is anyone familiar with the concept of thromboelastography? All right, that's so. Oh, we got one. Okay.
Well, I'm not familiar with it, but I guess.
Okay, what's the guess?
Like white blood cell, like stretchiness.
Close. Close. I mean, that's a great way. I mean, you're trying to get from the base words. Thromboelastography, which uses cytochalasin, is actually the means by which to measure your clotting. So when you have blood, so say you're hemorrhaging in the ER, and they need to figure out how to stop that hemorrhaging, they're going to take a small vial of your blood, stick it between two plates, spin it really, really fast, expose it to cytochalasin, and see how quickly you clot, so that they can administer the right drugs. These other two are more scientific tools. Phalodin, for instance, allows for the dense tagging of actin filaments so that you can kind of see things more specifically. But they are good terms to be aware of. Okay, so then we've also got intermediate filaments. Now these intermediate filaments are about 10 nanometers in diameter, so a little bit bigger than actin filaments. And they are actually a complex of, I believe, 8 dimers. And these dimers are actually dipolymers. Which means there are two polymers, two monomers wrapped around each other, and then we have 8 of those surrounded in a little tube-like structure. These are important for maintaining cell structure. They actually keep all the organelles that we had seen in those previous pictures in the right place. And they form the nuclear lamina, basically that nuclear envelope we were talking about. So that is the thing that actually sort of keeps the nucleus in place. A few disorders that matter, that affect the intermediate filaments, include things like arrhythmogenic right ventricular cardiomyopathy. Does anyone have any idea what that is? Okay, we got one hand shooting right up.
Is that when your heart the muscles are growing at a fast and large in the heart?
Close, so that you are identifying arrhythmia. So arrhythmia in this case is your heart not pumping synchronously, kind of a little stutter step. Very good. We got one bit of the details. That's one of our 4 words. Yes?
The right ventricle of your heart is pumping at the wrong time.
Yes, the right ventricle is in fact pumping at the wrong time because of what? Okay. Well, so a heart attack might induce it, but what that means, actually, heart attack is a great way of inducing it. Cardiomyopathy means, if you just ever want to do it, cardiomyopathy means that your heart muscle has been damaged and scarred. So what this is basically saying is you've got a stutter step right ventricle, one portion of your heart is not pumping right because the muscle itself has been damaged due to poor intermediate filaments. In fact, one last little physiological question. Where does the blood from your right ventricle go? You can shout this one out. No confidence. All right, we got some talking. All right, here, let me make it democratic. How many? So there's only 2 places it can go. It can go to the rest of the body. It can go to the lungs. Who wants it to go to the rest of the body? You got to vote for something. It's going somewhere, or it's just cool in there. All right, who wants it to go to the lungs? All right, it does look like democracy prevails in this case. The right ventricle pumps blood to the lungs. We've also got other things affecting the intermediate filaments, are things like desminopathies. Let me check my notes real quick just so I don't get this wrong. And yeah, that's just a bunch of heart and lung problems due to the intermediate filaments. There's epidermolysis bullosa simplex. In fact, I think it's like 1 in 100 people suffer from this, and it's basically a predilection to blistering. So if you blister easily, you might have a mild case of epidermolysis bullosa, I'm never going to get it, bullosa simplex, which is in fact a disorder of your intermediate filaments. And then laminopathies, problems with your nuclear envelope, are themselves intermediate filament problems. Okay. No. Finally, the last cytoskeletal structure I need you to know are microtubules. These are the biggest ones. They're about 25 nanometers in diameter. These play key roles in the transport of material across the cell. These are like your big superhighways, and these use motors, two molecular motors in general, dyneins and kinesins. And they also, if you're a bacteria or, I don't know, you got sort of your gut flagella, those are all, in fact, microtubules. They are made of a pair of monomers, alpha and beta monomers, that just go around in a big helix. Like I said, helices are important. So a few things, drugs and what they treat that sort of target the microtubules are colchicine. Oh, goodness, I should never try these words. Colchicine. Okay, there we go. Some people actually know how to say words. Some of us just bumble through them. This is actually a medicine that's given to gout. It also treats cancer, but everything treats cancer, as the other two drugs attest. And it binds the tubulin dimers themselves. Taxol, which is given in the case of breast and lung cancer, binds and stabilizes microtubules to prevent them from going forth and spreading. And something like vinblastine, which is used in leukemia, binds the dimers and prevents their polymerization. So these are a few good things to know. All right, now we're going to take an actual look at what some of these look like. So we had seen sort of this actin filament, this intermediate filament, this microtubule filament. This is sort of where they're located in a general cell. So you can see the actin filaments tend to make up the periphery. The intermediate filaments kind of go every which way and in fact encircle the nuclear envelope. And the microtubules going from a very key place. Does anyone know where they start? I leave that as an exercise to the reader. Okay, we got... No, they don't start at the cell membrane. They will end up there. Yes? Centrosome. That's a very good one. There it is, right there. It's a good thing to know. And you can kind of see it all here. It looks a little bit like these. These are some actual fluorescent microscopy images. So just so you remember, we aren't always just dealing with pretty pictures like this. Sometimes the actual biology looks a little messy. Okay, so the next...
That's really cool.
Oh, good. I tried. I didn't make any of these pictures.
I know, but they look cool.
Damn right. That's biology. All right. Moving on to sort of the next member, or the next organelle I need you to know, the Golgi apparatus tends to look a little bit like this. So you've got this little picture here, long parallel structures with some budding on them. You can see sort of this is what a nuclear envelope looks like. Here are what these vesicles kind of look like under a transmission electron microscope. It's budding off. And here's the actual Golgi apparatus. It's basically a bunch of stacks of pancakes. That's really the way you can identify it. As one of our colleagues in back here had actually mentioned, the Golgi apparatus is responsible for modifying and sorting proteins and phospholipids in order to tag them for transport. They basically say, okay, thanks, I've got a protein, I need to put it in the right place. Let me give it a label and make sure it gets where it needs to go. The Golgi apparatus is also important for things that are secreted either from the cell or to another cellular component. And that's done, as we had mentioned, through vesicles, these little budding things. So they've labeled the material, and they're responsible for kind of getting it to its last place. You can think of this as a packaging and processing place, PPP. Or you can think of it as like a shipping and receiving center. But on the topic of vesicles, and you let me know if I'm going too fast. I see that there are some people. Too fast? Okay, good. This is exactly what you should tell me. Don't be quiet. You just say, shut up for half a second. Let us write that down. So I'll give you just another few bits to write that down. Yeah, please. This is your class. If I'm not teaching as effectively, please just let me know.
The second part is vesicles, right? Not the actual
sorry, what was the question?
Golgi apparatus
These proteins are themselves secreted from the cell, or they're delivered to other cellular components. Yeah, the Golgi apparatus you don't want to be secreting. I'm sure that's a disorder of some kind. You had a question?
Which of the filaments are between cells and which of them are inside of cells?
Everything that I just mentioned was inside of the cell, except for a microtubule, which can extend beyond the cell membrane for cilia and flagella. Yeah, thank you. Yeah, great question. We're not quite there on like collagen and stuff yet, but we will one day. Okay, so now that we are coming leisurely to vesicles, vesicles are basically a little membrane-enclosed delivery packages. They themselves also have the phospholipid bilayer, and they are the main means of transportation of material in cells. So if you've got a bit of material that's got to end up somewhere, it's probably first being inside of a vesicle. Vesicles are also highly important to exocytosis and endocytosis. Exocytosis is the secretion of material from the cell, and endocytosis is the uptake of material into a cell. And that kind of looks a little bit like this. So we've got exocytosis up top, so we've got our Golgi apparatus budding off a vesicle. That vesicle on some kind of cytoskeletal structure is going to the cell membrane, merging with the cell membrane, and crapping out its stuff. And sometimes it is literal waste. Things like endocytosis, it goes the other way. So your cell says, oh, hey, that's some delicious looking material. I think I'm going to gobble that all up. And then it can be delivered a number of places, sometimes the Golgi apparatus. A few vesicles that you might want to know. We had someone over here point out that, in fact, a lysosome is a special kind of vesicle that just digests things using hydrolytic enzymes. Basically means you get something that you want to digest, you got to go ahead and stick it in a lysosome. And then another important one to know, perhaps just near and dear to my heart, are synaptic vesicles. These are the vesicles that exist at the synapses of neurons. So if no one's ever seen sort of a synapse of a neuron, it kind of looks a little like that. And here it is interfaced with another little synapse of the neuron. And they all keep these little neurotransmitters just right towards the edge, so that when they get a little signal, say another material goes and binds to the cell membrane here, this can be signaled to the cell, and it will then try to merge this thing and it's not great, but I'm trying to show that it will release its neurotransmitter into the synapse. So that's the way neurons communicate with each other, both electrically and chemically. Trying to draw a better picture than that. Okay, so that's pretty much what you need to know about vesicles and the Golgi apparatus. Now, one step removed, since we kind of have we've got proteins at the cell membrane. They got there through vesicles on cytoskeletons, first packaged and received at the Golgi apparatus. Where did those proteins come from? Well, it turns out they come from the endoplasmic reticulum. And the endoplasmic reticulum doesn't really look like this. Again, I can't stress that enough. Biology kind of looks a little more like that. It's densely packed, it's kind of wavy, and in fact all these tiny little studs, all these little black dots, are ribosomes. And these are the bread and butter of protein making. Here again you can see the nucleus. Here's the nuclear envelope, and then coming right off of that nuclear envelope is the endoplasmic reticulum, sort of signifying how related the two of them are. This is where cell membrane components and materials destined for transport, either within the cell or out of the cell, are made. So this is where proteins and lipids and carbohydrates are made. And in fact, like I had said, this produces proteins. Proteins are made in the rough endoplasmic reticulum, often called the rough ER. And lipids and carbohydrates are made in the smooth ER. So the rough ER tends to refer to all this ribosome-studded stuff. And then the smooth ER is stuff that doesn't have that. And then finally, like I had mentioned just a moment ago, the endoplasmic reticulum extends from the nuclear envelope, basically showing that it is super related to what's going on in the nucleus. So whatever the nucleus has going on, the endoplasmic reticulum wants to know about it and respond to its commands. Yes, question back.
So the proteins are produced by ER and both lipid and carbohydrate.
Are made in the smooth ER. Entirely correct. And in fact, we can kind of, while we're here, talking about the smooth and rough ER, get a look at what they actually look like. So right on top here is a transmission electron microscope. You can see studded here are those membrane-bound ribosomes. Here you can actually see a little bit of that mitochondrion. And then you got a few free ribosomes in the transition period between smooth and rough ER. And then you've got the smooth ER, which kind of looks almost like vesicles. And you have to be careful when you interpret pictures like this to recognize that there is a three-dimensional structure involved. You know, we always see things as 2D structures, but they actually represent many, many different things. So that's another thing just to keep in mind as you go ahead. So some of the details you need to know about rough ER, it's studded with ribosomes. It makes those proteins just like I was saying. And it's going to be, at least for this particular class, with a few exceptions here or there I'll point out, it's going to be the main kind of endoplasmic reticulum that we care about. It's not to say the smooth stuff doesn't matter. It just is to say that there's only so many hours in the day. But just so that you don't learn nothing about what the smooth ER does, smooth ER lacks ribosomes. That is definitionally the difference. And they tend to be within cells that secrete lipids, phospholipids, and steroids. Now, these are things like the ovaries, the testes, and the sebaceous glands. Does anyone know what a sebaceous gland is? You've answered one. Let's see. Is anyone new? Okay, way in back. Rohit?
Yeah.
All right. Sweat glands, exactly right. Yeah, it is a fancy way of saying your sweat glands. So if in your adolescence you were plagued by hormones and just the ravages of time, you have your smooth ER to blame for a lot of that. So, just a thing to keep in mind. All right, people don't seem to be too furiously writing. All right. So, actually, to this point, I would like to introduce just another segment that might be important. I've gotten this question a lot, a couple times at office hours, sometimes beforehand. What the heck does a biomedical engineer do with everything we just learned? What the heck do they do? Well, one example that I could think of, it involves the protein insulin. Now, insulin is a hormone that regulates glucose blood levels. So your blood sugar level has to be regulated by something. The reason we care about regulating your blood sugar level is sugar is a great source of energy, and we got to get it all throughout your body. So we got to make sure the levels that are inside your blood are regulated. Insulin does that. It is a protein. Where is it made? Shout it out.
Pancreas.
Oh, we got pancreas. I guess I meant in the cell. Where is it made?
Rough ER.
Rough ER, exactly. It is made, and how exactly does it exit the cell? Vesicles, exactly right. So knowing nothing more than that, knowing that it's made in the endoplasmic reticulum, the rough ER, and that we can use vesicles to pump it all out, biomedical engineers can basically trick E. coli cells into making as much of it as we want. When people around the world take insulin, they're basically taking it spit out from a bunch of E. coli cells. And so a biomedical engineer might be tasked with coming up with a kind of bioreactor, of parameterizing a bioreactor. And by that I mean determining the process, determining the actual chemical process of creating insulin within an E. coli cell. Maybe the sensors involved in that bioreactor, the materials involved, the flow rates involved, the quality system. You may need to ensure that it meets certain regulatory compliances. When the FDA comes up with rules, you, as biomedical engineers, need to follow them. And so a biomedical engineer needs to effectively extract that insulin. So this is the sort of thing that a biomedical engineer might do. This means that the 7 million people in America that rely on insulin every single day can actually rely on it, because people like you in this room know how to get from the endoplasmic reticulum to the Golgi apparatus outside of the cell insulin in any form we want it. So that concludes that little segment. Okay, moving on to the mitochondria. As a few people had pointed out, it's got a pretty distinct structure. It kind of looks like it might be a little organism inside of you. And that's because that was, evolutionarily speaking, the mitochondria used to be their own kind of thing. So if we go back to our ancestral prokaryotes, before we got ourselves nuclei and all sorts of organelles, we didn't have a mitochondria. About a couple billion years ago, mitochondria jumped ship. One cell or a couple of cells or a population of cells started eating up mitochondria. And it turns out they found that that was a great way to make sure their energy needs are met. Rather than trying to produce it themselves, plants, animals, fungi all decided to use mitochondria for a while. And in fact, plants also augment that with a bit of their own self-generated energy in the form of chloroplasts. The only kind of organism on Earth that lacks mitochondria are archaea bacteria, and I leave it to the interested reader, interested student, to figure out how they do that. How do they get their energy, if not from mitochondria? All right, if you know nothing else about mitochondria, what do you know? All right, it's good to know that would land. Yeah, if you know nothing else, you need to know that mitochondria is the powerhouse of the cell. But because you are in college now, we use some fancier words for that. In the mitochondria are responsible for generating chemical energy for cells via a series of oxidative reactions. And in this particular case, it is oxidative phosphorylation. And this makes a thing called adenosine triphosphate, or ATP. And this is the basic chemical fuel that powers just about all the activity in all cells everywhere on Earth. In fact, every single cell in your body. So, to highlight this, if you think about the mitochondria from now on, you don't need to just spout out as the powerhouse of the cell. It is in fact responsible for oxidative phosphorylation so that we can produce ATP, or adenosine triphosphate, so that we can have chemical energy to use for whatever purpose we want, be it the manufacture of proteins or their movement about the cell. And now if we take a look at sort of what that looks like in real life, and to highlight here, the mitochondria in this diagram is this little thing here, sort of that membrane-bound, membrane-enclosed protein. I believe we had, or not protein, organelle, someone had mentioned. Okay, if we take a look at this first thing, the first thing that gets done in the production of energy is that you've got to break down food into subunits. So if you get proteins from meat, or you get carbohydrates from bread, or you get fats from cheese, because you had your favorite pizza or something, you've got to break those down into amino acids, simple sugars, and sort of fatty acids and glycerol. That can all be seen here. This is usually done, at least in the case of multicellular organisms, not within a particular cell, but in organ systems. Next, you've got to break down those subunits, and this will be an important thing to write down, into acetyl coenzyme A. So the cell will then uptake these simplified proteins, or these simplified amino acids, simple sugars, fatty acids. And in the case of sugars and carbohydrates, perform a few more acts on it, such as glycolysis, to produce a little bit of ATP. Like I said, sugar is a great source of energy. And NADH, I always have to remember what this means. This is nicotinamide adenodinucleotide. You don't need to remember that actual word. You should just be familiarized that NADH is a precursor to ATP in a lot of cases, as is acetyl coenzyme A, because as these materials are broken down, pyruvate is also another way of digesting materials, especially proteins. It's also known as pyric acid. It's then brought into the mitochondria, where it is broken down further into acetyl coenzyme A. That acetyl coenzyme A is at the very beginning of what we call the citric acid cycle. You do not need to know the citric acid cycle, every single component, but you should understand its relevance. Oh goodness, we get out of here at 2:30, right? Oh dear. Okay. All right. Don't worry, we're going to make it. Okay, we've got the oxidation. So finally, in the mitochondria, we get the oxidation of acetyl coenzyme A into water and ATP. So again, if you learn nothing else about the mitochondria in this class, be aware that it is responsible for oxidative phosphorylation, and it produces adenosine triphosphate, ATP. Finally, when we start to speak about ourselves consuming oxygen or producing carbon dioxide, we're actually speaking on behalf of our mitochondria. You yourself do not consume oxygen. Your mitochondria does. So just be aware of that. Humble yourselves every now and then. Then finally, coming to the heart of the matter, the nucleus. Nucleus, like I said, doesn't really look like that. Looks a little bit more like that. Can anyone identify what those little darker blotches are? You can shout that out if you know. Nucleolus, exactly right. It's responsible for making the ribosomes. That's actually where they get made. But the nucleus in general is a membrane-enclosed organelle found in all eukaryotic cells. So eukaryotic cell is the key term to know here. It's the phrase that pays today. Some cells in our body have none. For instance, red blood cells do not have nuclei. This makes the reproduction of red blood cells difficult, which is why hemorrhage is such a catastrophic injury. And some have a lot. Some need to do a lot of things. Things like our skeletal muscles actually have more than one nucleus per cell. So you can have one, you can have none, you can have a lot. Biology, turns out, is a big tent and can allow for a lot of variation. And finally, the nucleus contains and maintains genetic material of the organism, and it controls its expression in the cell. So it contains and maintains. Both of those are important. It not only holds the genetic material, but it makes sure it's safe. And then it also controls the flow of information into and out of the nucleus. What matters to the nucleus, just like the mitochondria, if you get nothing else out of it, you ought to associate the nucleus with the genetic material. There we go. That's exactly what I'm looking for. Again, shout. Do not be afraid. What's that? DNA. Oh yeah. You're gonna know something here in a bit. Sorry, what was that? Was that a question or? You're anticipating me. All right, we're all. I apologize, and if I am rushing, it's just because there's so much to learn. Okay, so the genetic material is what's important. Yes.
Sorry. What do you mean by maintaining the genetic material?
We will learn that in a bit. But that is a great question. So nothing lasts forever. It's got to be actively maintained. If you want something to stay fresh and alive, you've got to expend energy to do that. That's sort of what I mean. All right, so the genetic material, it is important to know this about the genetic material. I believe it might be written down on your notes. It is the inherited set of instructions that guide the development of an organism, encoded molecularly in all living organisms by deoxyribonucleic acid. And I think maybe on your original notes I had a typo there. I apologize if that was the case. So what do we mean by a phrase like this? There's a lot to unpack there. Well, it must be inherited. Genetic material comes from another cell. All cells come from cells. That is the basis of our cellular theory. It's an set of instructions, which means that it's a code that needs to be understood and acted upon. So we've got, a whole bunch of things that we as a cell want to make: proteins, lipids, cholesterol, and we need to know how to do that. Because our genes are going to be with us from the cradle to the grave. From the moment we bud from one cell into two, to the final apoptosis of our last living, breathing cell, our genes will help guide us. It is encoded molecularly, in this case, sugars, nucleotides, and phosphates. And finally, The actual molecule that we all care about is deoxyribonucleic acid, DNA, which will actually be the subject of our lecture next time. Does anyone know what this is? What this? Yeah.
It's an X-ray image of the spectrum.
It is an X-ray diffraction pattern. But yes, you're entirely right. I'm just helping. It is an X-ray diffraction pattern of DNA, the helix structure. Does anyone know why this particular picture is famous? All right, we have it right over here.
First one.
This is the first one. Does anyone know who made it? Oh, okay. Let's see. Who's new? All right, here we go. Rosalind Franklin was actually the advisor of the person who took the picture. But she is responsible. Yes, she is the uber genius that knew how to conduct this experiment. Does anyone actually know the PhD student that took the picture? Yeah, no one remembers the PhD student. It's a fellow named Raymond Gosling. But if you're going to remember a person, try to remember Rosalind Franklin. She is, in fact, responsible for us knowing the double helix. I'm going to give us no break at all, and we're going to now turn to 3 quotes triangulating. I'm sorry, I just love this aspect of the class. I will bore you to tears with it. Okay, so today we have introduced to ourselves biology, cellular biology. Niccolo Machiavelli, the person who wrote The Prince and responsible for the phrase Machiavellian, had the following phrase. Would anyone like to read it out loud and be very princely or princessly? Commanderly? I'll read it. All right, if you're all bored with it. Okay. So, it ought to be remembered that there is nothing more difficult to take in hand, nothing more perilous to conduct or more uncertain in its success than to take the lead in the introduction of a new order of things. Because the innovator has for all enemies all those who have done well under the old conditions, and lukewarm defenders in those who may do well under the new. This coolness arises partly from fear of the opponents who have the laws on their side, and partly from the incredulity of men who do not readily believe in new things until they have a long experience of them. So if, when you are introduced to new material, you find it hard to uptake, that's okay. It's natural. It is an unfortunate reality in life. Okay, does anyone want to read this next quote? This is one of my favorites. No one wants to read nothing. I'll blow through these today. I am, unfortunately, coming up against it. This is from Robert Hooke. This actually comes from his famous publication, Micrographia, and this is the very first instance of the word cell. I took a good, clear piece of cork, and with a penknife sharpened as keen as a razor, I cut a piece of it off, and thereby left the surface of it exceedingly smooth. Then examined it diligently with a microscope. Methought I could perceive it to appear a little porous. These pores, or cells, were not very deep, but consisted of great many little boxes. I no sooner discerned these, which were indeed the first microscopical pores I ever saw, and perhaps that were ever seen. For I had not met with any writer or person that had made any mention of them before this. But methought I had, with the discovery of them, presently hinted to me the true and intelligible reason of all phenomena of cork. He recognized in seeing these cells under his microscope that he understood something very, very true about the phenomena of cork, of a living organism. And in fact, this is a drawing. The drawing made 352 years ago in this book of what he saw through his microscope. This is the very first image of a cell we have. And it's as recent as 350 years ago. There are park benches in Britain older than our knowledge of the cell. So keep that in mind. And finally, one final quote just about biology, in case you ever want to understand it. I venture another and perhaps equally reckless generalization. Nothing in biology makes sense except in the light of evolution. Subspecies evolutionists. This is from Theodosius Dobzhansky and his essay, Biology, Molecular and Organismic, basically saying that if you want to understand why a cell, an organism, an ecosystem does something, be aware of its evolutionary history. So if you want to know why the mitochondria does what it does, try to figure out how it got there. Why did the endoplasmic reticulum look the way it does? What were the forces shaping it? And so long as you keep the light of evolution shined completely upon it, you will have some sense of it. Okay, so now for something completely different. I will pass back here after class. Please come get them, or I can just give them to you next time. I like a few things about your signatures. We had a flower child in the class, an assistant to the regional manager. We had a couple future engineers. And then some of your signatures just look great. This is a perfect B if I've ever seen one. I like Nichols. Nickel, where are you? It's, somewhere. Just believed that a single name was enough, like Prince. So it's pretty good. And now just a few unsolicited opinions on the use of pencils or pens. I would recommend that you always use a pen. One of the nice things is pens don't erase, they don't go away. So that when you want to refer back to these notes in a month or a week or a year, the graphite, if the papers rub up against each other, will start to smear and disappear. So I say invest in a pen that you like and use it. And instead of erasing things, try just to strike them out with a single strikeout. I'll understand you don't want that graded, or that's not what you intended, but sometimes you may have had the nugget of an idea or a turn of phrase that you liked, and if you just violently get rid of it, you'll never know what it was you meant. Okay. So let's, here at the tail end, work our way out. What is this little thing, and what does it do? I'm just going to start right over here. Erica, what is this thing, and what does it do? That's the nucleus. Fantastic. It has all the instructions and controls their expression. Coming from the nucleus is what? What branches off of the nucleus? Well, there's the nuclear envelope, which separates the nucleus from the rest of the cell, but a lot of people said endoplasmic reticulum. That's entirely right. That creates proteins via ribosomes, lipids, and carbohydrates, and that is its function. It then sends those proteins somewhere. Where does it send them? Or how does it send them first? Vesicles. Fantastic. Those little things right there. That transports materials through the cell. Where? What else uses a vesicle? I didn't get my timing right. A special kind of vesicle is the lysosome, which is responsible for digesting things we don't like. You had all mentioned the Golgi apparatus. This makes some vesicles themselves, and it packages and processes them, making sure everything gets to where it ought to. What is that little salty jelly that's in the middle of the cell? Cytosol. Very good. That is, in fact, all this movement takes place in the salty jelly of the cell via the cytoskeleton. And then finally, we've got the mitochondrion and the peroxisome. And this is where all the energy comes from. And sometimes those peroxisomes, when we can't digest simple things, will do so very brutally. And separating all of this from everything not living is the cell membrane. Now, why do we care about any of this? A lot of medical problems arise when the cell or tissue function is altered. So when we start to talk about problems like diabetes or cardiomyopathy, we are at the very least talking about a bunch of problems with the cells. So how can we tell? That's a question that you're going to need to think about for your projects, knowing what you know about these systems and subsystems. And then why might we want to tell? Sort of keep that in mind. Now, finally, I'll leave you with one last thing today in the last remaining 5 minutes. This is today's number to know. On Monday, it was the 16th anniversary of 9/11. On that day, we lost 2,974 people. Of that number, 1,641 victims have thus far been identified. The vast majority of which have been identified via biomedical means, biotechnology, usually DNA sequencing, things like dental x-rays, and fingerprint analysis. All of this is made possible by biomedical engineers just like you developing new technologies. So if you want a reason for why you ought to do what you're doing in this room, think about one of the latest victims identified, a man by the name of Matthew David Yarnell, whose family now knows, who now has a sense of comfort brought in only the way that biomedical engineers can do. Said of Matt by one of his friends was he was the kind of person that you could talk to about everything. Career, finances, love, future. He was always there for his friends, willing to listen and give advice and a warm hug. biomedical engineers touch on the human condition in a way a lot of engineers don't get to. A lot of electrical engineers won't get a story like this. A lot of mechanical engineers, sure, they may save lives with safer cars, but we will actually get to touch people in the biomedical condition. And with that, hopefully we can have a little bit more love in the future, and I will see you all next time. Thank you. If you would like to get your letter, you can come up here or I'll bring it next time. Sorry, oh, yeah. Absolutely. Right. There you go, I got it in alphabetical order.
Are the lectures always Tuesday?
They are Monday and Wednesday.
Sorry, the after 7 o'clock one.
That's only happening Tuesday and Wednesday. This week.
And then the bioethics one that you did?
Every other Tuesday. Truman Cole, what's last name? Stovall, that's what it was. Stovall. I know I've got it, there you go. We may need to take this outside, but yes. Right shore, okay, right shore. We will get it. There you go, Annika. Yes?
I just want to say thank you for the email, and
Oh,
I
no.
think all
Okay, thank you, take care. Yes? Suma. Okay. Suma. If you see it, you just let me know. Suma. There you go. Thank you. Yes. Okay, I will take those. Thank you very much. Yes. Okay. Samantha, sorry, what? Oh, you're going to be the fine. There you go. The future engineer. All right. Trip. That'd be, I remember this one. I remember Thaddeus. There you go. Thank you. Michael Rakowicki. Here you go, Michael. I just saw yours. If we get kicked out of here, hopefully we don't, but there. Oh, nope. There you go. Thank you. There you are. That was your. Yep, very good. Alex Socha. There you go. Yeah, you had one of the ones up there. Yes. Rakitha, what is your last name?
Nimagata.
Yeah, Nimagata.
There's another Nimagata in this class.
There is indeed. Yeah, Shriman. Okay. Looks like, when does your class start? Okay, well then we'll get out of here. Okay. Yes, so we will take this outside if you all don't mind.